Okay, hello everyone. Can you hear me properly? Good. Hi. First of all, I'd like to thank the organizing committee for giving me this opportunity. My name is Gaurav. I'm a PhD student with Juncker Lab back in MDC Berlin, and today I'm going to talk about one of my projects called predicting developmental states using transfer learning. So we all know that the single-cell genomics has revolutionized the way we study biological processes, as it allows for the capture of transcriptome across different developmental stages, organs. Yet these techniques only provide static snapshot across these biological dynamics, revealing a cell state in a gene expression space. And many computational approach has been developed to reconstruct this developmental trajectory based on the similarity of transcriptomes. However, such methods are descriptive in nature and unable to interpolate and extrapolate the missing data points. So also it is quite unknown to which degree these cell trajectories are predictable. So what we want to predict a cell state, learn from one lineage about these states, and how predictable is this early development specifically, because we work on the early zebrafish development. So like learning from one data and predicting the other, like the cell state, this is what a transfer learning is all about. So what transfer learning do is that it store a knowledge while solving one problem and then use that knowledge to apply on a different problem. And there's been many methods being developed on this concept. For example, on data integration and then predicting the states across diseases and species. The one, mostly these methods are used by a supervised way. For example, you use this deep learning method so that you can compress this information and put it in a latent space where it's very easy to transverse and manipulate. So what I want to predict the developmental state using transfer learning. So what you're looking at is this beautiful single-cell RNA-seq data of zebrafish embryogenesis across the first 12 hours of development, where each point represents a cell. And this is a very cool data because it includes this emergence of a cell diversity at gastrulation, which is the time where the non-linearity hit in the system, and which is very powerful test case for the inference of transcriptional cell state. So what we want, I can't have a pointer, but on the right you can see there is this emergence of these 3 germ layers. For example, this mesoderm, which give rise to the internal parts, whereas, the external parts, and on the left you can see this nice internal ectoderm layer. So I'll be what I did is I tried to learn, for example, from 5.3 hour post-fertilization cell state, and then trying to predict the 12 HPF, like these 12 hour post-fertilization cell states. So we started with a very simple approach of a vector arithmetic in a gene expression space. One should remember that I'm using all the 18,000 genes. I'm not looking for any highly variable genes. This is pure data set. So in the pink, you can see this mesodermal layer, from which I'm learning this vector by taking only a difference and then substituting in on a 5.3 cell state to get these these cell state information. And I'm here comparing the real and the predict data, where you can see that the R square is quite impressively remarkable, that it's around 0.98, whereas one could expect that this is a simple vector arithmetic on a mean gene expression, so it can't count for the increase in the variance. So we thought of using the previous method, which works on a data integration. For example, I think everyone have shown this method from the lab of Fabian Theis, where they use these transfer learning approach to predict the perturbation and the drug responses across cell types, where what they did is they compress this information in a latent space and then learn this simple vector where you can transform this vector to these what you want to predict, and then again come back decoding this information in the gene expression space. And also, recently there is also one work on drug responses, which is pretty cool. So we use this method on our this zebrafish data set so that we can recover the variance. But unfortunately, it also doesn't work for us. Although we able to have a capture the mean gene expression as previous vector arithmetic, but there is no great success we got. So what I thought is to come up with a custom variational autoencoder model, where now what I'm using is this new normalizing flow, which, okay, so wait. So this is two-step process. First, you learn the latent representation by this modified variational autoencoder that I designed using these new reconstruction laws, and then some regularization. This is what you need. But most importantly is this second approach, what we call normalizing flow, where it explicitly learn the increase in the variance, which is quite good. And this is, so for example, I'm giving you this example where what I want is, I want to transform this green square into a blue rectangle. One cool thing is that it has the same area. So with this approach, I learned the map on one lineage, and then trying to use this map to predict the cell state, and more explicitly, the increase in the variance. So on the bottom, as you can see, there is this a nice increase in the R square for standard deviation. But if you can appreciate for the first two principal components, that now I'm able to capture these real data also. So this was good. So what we did is now apply this method, like considering just swapping the test and training data, and also these layers, and also look backward and forward. So the question mark represent what I am going to predict. So overall, as you can see, I'm like nicely predicting the mean gene expression, but somewhat we also predicting the standard deviation. We should not expect like the correlation of 0.99 for a standard deviation, because these are all different cell types. So we thought if it's working on zebrafish, then it should also work on the different systems. So we looked into hematopoietic systems. So I considered this data set on mouse hematopoiesis from this paper, the Larry paper, if you remember, which also used this information about the barcode. So I only concentrate on these monocyte and neutrophils lineage. As you can see, when I apply this method, I got a very poor prediction with these, by considering all these different scenarios. And we thought, why is this happening? So we then looked specifically for the prediction of monocyte, and then we found out that there are these very highly expressed lineage-specific neutrophil genes that is causing a problem. And then we thought, okay, so these genes have a very detrimental effect on the overall predictability. So what if we remove these genes? We should expect that the prediction goes up, and this is what exactly happened. So, but as you can see, you can reach the prediction about 0.7, 0.8, but it's not like the embryonic development where you have this nice correlation of 0.99. So we thought this is because the fundamental differences between the zebrafish development and the hematopoietic system. That's why we are getting this results. Okay, so then we thought, then we reasoned the degree of the predictability is based on the genes that are changing in a concordant manner in your training and your test data sets. So we just looked at the training and the test data set. Here I'm not doing any predictions, but see what is going on there. So on the left, you can see the result for the development. So, and these are the log fold changes between the early and the final, this 12-hour time point. We see that there is this high correlation among the ectoderm and the mesoderm lineage compared to what we are looking for in neutrophil and monocyte. So this says that the degree of predictability is quite depend upon the mutual information between this lineage, which I quantify using this metric called mutual information, where you can see that it's a very high mutual information. And I think I was very quick. And finally, what I want to see is that why is this happening? Why? What are these concordant genes? What are these biological programs it's encoding? So what I did, I look for these genes which have a very low absolute error. What it means that these are the genes I'm able to predict very correctly. So I use these genes, and I do a geotome analysis, and I found that these genes are mostly regulating the cell cycle and the metabolism, which makes sense in the early development. So I think with that, I would like to finish. Yeah. So the most important thing I'd like to share is that the predictability depends on the shared information between your training data. So that's why we can't able to do very well with the hematopoietic system. And these can be improved by having more information. For example, as I think, using this gene regulatory modeling, or you can have more information about like these accessibility information in terms of attack. So if you can make the model together with this unsupervised approach, but including this gene regulatory information, I think it can yield to a more predictive power. So with that, I'd like to thank my supervisor, Philip, and together with Bastian for directing or giving me this opportunity, and also my funding sources, and also you for your kind attention. Yeah, with that, I'll stop.
Thank you very much for the talk and for being right on time. Yeah. Fantastic. So we have time for questions, please. So maybe I can start with a quick one. Sure. So we are performing some prediction here, some major extrapolation, possibly, right, of cell states. What do you think are the limits of that, and how can we estimate?
So what was first?
So we are extrapolating, right, when we're making these predictions. What are the limits of this extrapolation? At which point we can say we are able to make this prediction, and when can we say we just don't have enough information to see how the cells are going to evolve, develop?
I think this is where everything is here. So it's totally depends on on the mutual information. So what it means is, if you have a shared programs, if the cell does not commit to be a very specific cell type, this is what I'll conclude from the development. Because this is where I work. So for till 12 hours you can predict, but also I apply this to 15 hours and 24 hours on this another zebrafish Wagner data, where it goes down so bit. So as soon as the cells start becoming more specific, I think you lose this predictability.
I have a similar question going along sort of limitations. You know, the beauty, I guess, or maybe simplicity of those vector arithmetics in latent space was that it's very constrained. So thereby you could argue maybe because of this regularization, potentially you gain a little bit more predictability if it works. And you could easily test when it works. Normalizing flows are super flexible. What type of metric could you use a priori to really quantify, or maybe to constrain it to make sure that you pick up those cases right away?
Exactly. So here I'm using the simple planar flows. But for example, there are, if you know, these coupling networks where you parameterize these flows using the here. So you use this function f. So here you can use the coupling information. It's all about what the other feature is doing, and it can help. You can have a function depending on that parameter. It's like the condition. I think this goes to this question of disentanglement, where I think if you use like disentangle everything, I think you lose the information about your neighbor features. So I think in a way that you can somehow use this this kind of normalizing flow where you also using the neighboring feature information. I think this can go. Yeah.
All right, let's thank our speaker again. The next talk is by Anita Térmeg from LMU Munich, who's going to talk about cross-species comparison of gene regulatory networks.
Thank you for the introduction. It's a pleasure to be here. Today, I would like to talk about gene regulatory networks and how to compare them across species, in my case, across primates. So first of all, why is it interesting at all to do such comparisons? We have a double benefit this way. Firstly, we learn something about the evolution of these species, and also we learn something about the networks themselves as systems, because evolution serves as a kind of large-scale experiment where these networks get modified and then we see the outcome. But such cross-species analysis also come with new challenges. One of the critical steps of network analysis is the detection of network modules. It's very useful to think in terms of these modules because they are often more conserved or more closely related to a cellular function than any particular gene alone. But to assign these modules, it often involves arbitrary cutoffs, arbitrary assumptions, and this can introduce biases in case of a cross-species setting. Another issue is the statistical power. Once we have these modules, it will be very intuitive to just compare how much they overlap between different species. But if we observe only a small overlap, it's very difficult to know if it's really due to divergence or just due to the lack of power. So my goal was to develop a computational pipeline that overcomes some of these issues and at the same time apply it to a primate single-cell dataset taken from a neuronal differentiation experiment. This experiment was performed by Johanna Geuder, a PhD student in our lab. She differentiated primate iPS cells into neural precursor cells over the course of 9 days. And she took samples at 6 different time points that were then sequenced using single-cell RNA-seq. So this way, we had single-cell data from 3 human, 2 gorilla, and 4 cynomolgus clones. Here you can see an overview of the pipeline that I developed. It starts with the normalized expression counts, and in the end, it outputs a ranking of the modules based on conservation. The most interesting ones can then be analyzed in more detail. So the first step is the network inference. The network inference algorithms essentially look at gene-gene pairs and try to estimate the strength of the regulatory interactions between them. I tested 2 different inference methods, and in the end opted for GRM Boost 2 that uses decision tree learning to predict these interaction scores. The next step is the module detection. After the network inference, I had separate networks for each clone, and here I combined them into a single consensus and did the module assignment based on just this consensus network. For the assignment, I selected putative regulators or putative hub genes. These were transcription factors that had an annotated motif and that were variably expressed over the course of the differentiation timeline. And then I built initial modules around these hubs by incorporating all target genes that the interaction score higher than 0. So essentially, all target genes that were connected by an edge to this hub. But these initial modules were often too large. They contained up to several thousand genes, which is obviously not biologically meaningful anymore, so they had to be pruned. Again, I tried different pruning methods, and one of them that's based on knee point detection proved to be the best. For this, I filtered the target genes based on two characteristics. First, how strongly they are regulated by the hub gene. This is the hub target interaction score. And secondly, how well they are connected to the rest of the module. This is the intramodular connectivity. I ranked all target genes based on these two characteristics, calculated the cumulative sum curves, and then identified the knee points of these curves. And in the end, the targets that were above these knee points in both curves ended up in the final module. So this method has the advantage that it's an adaptive filtering, so we don't have to predefine, for example, how many genes we want in the final module. And it also allows for overlaps between the modules. So it successfully reduced the module size by an order of magnitude, and it also increased the proportion of direct targets in the modules. So once we have these final modules, we can go on to study module preservation. I used the module assignment from the consensus network in all of the clone-wise networks, and then compared how preserved these modules are between different pairs of clones. And for this, I adapted the concept of Z connectivity from WGCNA. It quantifies how similar are the interaction patterns between a reference and a test clone. And I extended the original definition to be also applicable to overlapping modules and directed interactions. So this way, I don't calculate overlaps between module member genes, but I compare these interactions in a more quantitative manner. And after this step, I ended up with preservation scores between all clone pairs. They can be summarized in a matrix like this. Some of them measure preservation between clones from the same species, those are the blue ones, and some of them measure preservation between clones from different species. In the next few steps of the pipeline, I summarized these Z connectivity scores into a measure of cross-species conservation. So in principle, if we assign these modules well, we expect them to be highly preserved within the same species and somewhat less preserved across different species. This is how the scores look for an example module, SMAD4. It's an important regulator of neuronal differentiation. And overall, the within species scores are indeed higher than the across species scores for all three possible species pairs. Here, I show you the same comparison for all modules. And again, the across species scores tend to be lower than within species scores, at least for the gorilla cynomolgus and human cynomolgus species pairs. For the human-gorilla comparison, the difference is much less visible. But this is exactly what we expect based on the phylogenetic tree. Because in terms of phylogenetic distance, the gorilla is much closer to the human than either of them to the cynomolgus monkey. So in general, for distant enough species, the hypothesis seems to hold that within species preservation is higher than across species preservation. But the effect size varies from module to module. For example, for the Apple module, the differences are minor, if there's any, And for the bottom module, in some cases, the differences are very pronounced. So to quantify these effect sizes and compare them, I used t-statistics. I performed a t-test for each module and each pair of species and used the p-values from these t-tests to rank the modules from most diverged to most conserved. Here you can see the results on these 3 volcano plots. They correspond to the 3 rankings. And for example, the famous pluripotency factor nanog falls quite low in all of these rankings, which makes perfect sense since it's a very essential gene. I also identified the top 5 most diverged and top 5 most conserved modules for each pair of species. Here you can see one of the most conserved modules, CREB3L2, and one of the most diverged modules, RXRA, in more detail. And the Z-connectivity scores for the CREB3L2 module are quite similar for all clone pairs, which shows that this is a conserved module, while in case of the RXRA, the human cynomolgus and gorilla cynomolgus scores are much lower than the rest, which indicates divergence between the great apes and cynomolgus. After we identify such interesting modules, we can do some downstream analysis on them. Our future plans include comparing the overall expression profiles of these modules using the concept of module eigengenes. And we are also working on a CRISPR-I screen where we are planning to repress transcription factors in iPS cells and see how these networks behave under perturbed conditions. So to sum up, I developed a pipeline to compare networks across species that includes dynamic module pruning, an adaptation of the Z-connectivity statistic, and a new approach for quantifying cross-species conservation. And I applied it to a primate single-cell dataset and found that one of the most diverged modules during early neuronal differentiation is RXRA. So at the end of my talk, I would like to say thank you to my supervisor, Ines Hellman, and the entire Hellman-NR lab for their support. And also thank you for your attention. If you have any questions or suggestions, I'm very happy to hear them.
Thank you very much for a very interesting talk. We have time for questions, please.
Hi. Yeah, that was very fascinating. I was wondering how many cells you need to derive such a module.
Well, in our case, we had around 500 cells per clone. So this is enough to construct such a co-expression network. But if we have more cells, then it's also a good strategy to first do pseudobulk samples and then do the network inference, because this can really decrease the problem that comes from dropouts in case of a single-cell data. So in our case, we didn't have enough cells for this, but in another data set where we do, I would recommend to do that.
Okay, thanks.
Oh, hi, thank you for your talk. I was curious, are you only considering genes that share the same names? Because if you're looking across species, you're probably looking at different gene sets. Are you accounting for homologs? And if you're not, is that potentially biasing your scores towards being higher within species than across?
Thank you. This is a very good point, because the basis of all of this is to have match the genes or match the nodes between the different species. And for this, we used one-to-one orthologs from the Ensemble database, and we also added on top of it genes with the same name in the GTO files.
Okay, thanks for the nice talk. So I also have a kind of related question about the genes. So as I understood it, you use a specific number, not specific, but a few number, like 100 or 200 genes, right? So, and then how does the genes that you build your network with affect the module conservation? For example, if you build your network with transcription factors to get more divergence than, I don't know, like other genes, does that have any effect?
Yes, of course. It's very important to select the hub genes well, because this is really something we predefine. We don't take this from the data. Or maybe to some extent in this case, because I took the variably expressed genes. But essentially, I considered only transcription factors. And the rationale behind this is that we believe these hub genes to be transcription factors and then regulate other genes by binding to transcription factor binding sites and then repressing or activating their target genes. So there's this underlying biological concept.
All right, let's take our speaker one more time. Thank you. And the last talk for today, for this session, is from Maren Büttner from the DZNE Bonn, who is going to talk about swarm learning for single-cell data in human.
All right, thank you very much. So I know it's me between you and a coffee break. I'm not going to make it quick. All right, let's have a journey towards swarm learning. So when we look at medical data and the healthcare sector in particular, we know that this is disease-centered. And at the same time, we collect multidimensional data from imaging to omics data, single-cell omics even, or we collect data from these kind of wearables that me or others have on their wrist. Yet our data is very decentralized. It is stored at different locations and entities. We have high bars behind the privacy. So all of your data is very sensitive. It should be protected. And usually it's very siloed. So no one really has access to it, even if they wanted to. We have this multidimensional data. This is huge information. And we could use machine learning methods to leverage all of this information. Yet, when we want to come to true precision medicine, as Christoph has showed us this morning, we need machine learning to do this, and we need data. So, when we now go on and use machine learning on our data, I would like to take you on the journey towards decentralized learning now. So imagine we have these 5 centers in Germany who are sufficiently large and have sufficiently amount of data so they could actually start to do modeling on those, let's say training a machine learning classifier. So we could do this in a localized fashion, so every center just does their own model, they have their data, they have their classifier, voila. So there are a couple of issues with that. First of all, the data sets that every center has is still limited. We do have some local bias because the cohort that we collect in Bonn might be different from the one in Berlin. Local classifiers might be have low accuracy. Every center is disconnected, and these things are generally not generalizable. So, and with that, there is no real-world scenario possible. So let's hope all of these centers become aware of these issues and want to collaborate. So we could do this in a kind of cloud-based fashion or centralized fashion. So for instance, the Munich Center volunteers to take all the data, train the models, and this is what we now call centralized learning. So you have one entity that you sufficiently trust so that you donate your data. So this already gives us a couple of solutions. Data sets are much larger, we have less bias, we have a better accuracy, data is connected, generalizable, and the whole thing might be one step closer towards real-world scenarios. Still, when we look at human data, we have to comply with data protection regulations, GDPR in Europe. So let's imagine for a second that we don't need this centralized entity. We don't need to care about GDPR. And that is the idea of swarm learning. The data stays at every center, and everyone trains jointly one model, exchanges parameters, and gathers the same knowledge, the same insights without data sharing. So we have the same access to large data sets, better accuracy. We use blockchains to, let's say, ensure equality among the partners. We don't need data movement anymore. There is no central entity that is required for merging parameters, and everyone keeps the data sovereignty. So our goal is to share insights, not data. So the swarm learning principles are, we can integrate any private medical data from any owner worldwide without data sharing, without violating privacy laws. We can use decentralized machine learning methods, and we're pretty flexible with the methods that we use here. We unite edge computing, so that means every center does their own computing, and this is blockchain-based and a peer-to-peer networking and coordination, and that helps us to maintain the confidentiality of your data without any central coordinator. So we have set up the swarm learning for single-cell RNA sequencing data, and we chose the topic of data integration. We have set up a real swarm learning network between the LMU Munich and the DZNE in Bonn. So for data integration, we employ a federated version of the SCVI. And SCVI is one of the top-performing tools right now for data integration of single-cell RNA sequencing data. If you want to know details about that one, you can ask Josef about this. So what we construct is a latent space using an autoencoder. So in this autoencoder, we can use a distributed fashion. And from the autoencoder, all we need to know is this kind of latent space, which we can then use to compute an integrated embedding. So for swarm learning and for any type of distributed learning algorithm, we need to share the model parameters, which are, in our case, the weights of the autoencoder. We have two different versions for this. So in embed SCVI, we have weighted merge of parameters, whereas in the general swarm learning setup, we have equal merging. So independent of the data set size, we simply average all the parameters. One word about the model, I have to thank Mo Latvolahi and Sergei Rybakov from Fabian Theis's lab, who have developed the model. So we set up to do this with a, let's say, small example, which I think I have understood quite well. We published this in the benchmarking dataset integration paper, which Maria has already mentioned. So we have the lung cell immune data with 32,000 cells, 17 cell types from 3 different sites. So we simulated a swarm setup with 3 different sites that have collected these data independently. So now we can compute a data integration, and now I'll show you 3 different UMAPs colored by either cell types in the top row or batch in the bottom row. And what you can probably appreciate is that they all kind of look the same. And as I don't advise anyone to look at complicated UMAPs, I rather advise you to look at certain metrics that tell you how well you integrated your data while you preserved your biologically relevant information. So, and with our, let's say, small and quick tests that we did here, we see that swarm learning embedding SVI was ranking best among the tested models. So let's scale this up. we go for the lung cell atlas, human lung cell atlas. So, and here I would say there's the one atlas, one PhD hypothesis right now. So I have to thank Lisa Sigemer from Fabian Theis's lab for giving us the data with almost 600,000 cells, 61 cell types, 166 batches, so this is massive. They were collected at 14 different sites, and we simulated these 14 different sites at both DZNE and LMU Munich in our swarm network, and I will show you the results, let's say just looking at the UMAPs for our data set integration, and you see that these look astonishingly alike. When we now move on to look at the metrics and quantify how well do we do with batch integration and biopreservation, we still see that the swarm network is doing slightly better with the data, cell type preservation. So these are all still preliminary results. We have to do a lot of optimization and quick text, but this is very promising for us. So when we move on to look more into human immunology, we are faced with different data modalities. So as in immunology, we usually look at surface markers using flow cytometry, mass cytometry, or site-seq methods. They all cover different feature spaces and have different throughput. So, but when we look at flow data, this is one of the major endpoints when you look at clinical data or medical disease prediction, be it tuberculosis, AML, or other immune diseases that you can predict from flow data. So we could automatize the entire workflows around flow data, plus doing automatized diagnosis tools, so assisting doctors in the diagnosis so that they could spend more time with their patients. And we could achieve this using swarm learning. During the, we used the occasion of the current pandemic to establish some kind of global network using connecting everyone who has, let's say, collected flow or mass cytometry data from COVID-19 patients. So we already teamed up with lots and lots of partners who see the necessity for global collaborations. And I hope that you could join us at some point too. And with that, I'd like to thank everyone who was involved in this amazing endeavor, LMU Munich, LMU AI, our partners from HPE and the DZNE. I take questions, and if you have even more questions, come to my poster number 15. Thank you.
Thank you for a great talk. We have time for questions.
So thank you for the great talk. It seems that from the SCIB metrics on the federated integration, especially the biological preservation seems to be improved with the federated approach, if I understood correctly. Do you have any explanation why this is the case?
Yeah, thank you for the excellent question. So the metrics that we have been computing so far is how well can we recover the cell types through clustering. So and apparently this is still quite good. I'm still cautious about the result because the worse you integrate, the better usually this kind of biopreservation is and vice versa. So you can always have the tendency to over-integrate, and this is not the case for the swarm learning. So it does not over-integrate. We still have to evaluate how well, let's say, if it's not under-integrating. So there's always this kind of balance that we have to take into account, and that's why we have to evaluate these two.
Thanks for the great talk. I was wondering if you share the network weights. Of course, this is not explicit information about any human data, but I mean, you trained the network with the human data. So I mean, I think it's possible that there are traces of the data in the weights. And I was wondering whether there's any kind of proof saying that there is a non-identifiability if you only share the network weights, or is it just hard because no one knows how to do it?
That's a very interesting question. We have colleagues from the CISPA in Saarbrücken who actually do these kind of, let's say, attacks. How identifiable are people and data from these kind of latent embeddings in networks? I would say this is still ongoing research, so far as I can tell.
All right, thank you.
I also do have a question, if I can go ahead. Hopefully a quick one, great talk. When it comes to dealing with data from different centers, we can have within center or within dataset batches. How well those are handled in context of the swarm learning? Because it seems that we have only one single global covariate or factor that we specify as our batch, but it's actually very likely that in clinical settings we've got within a study or within center batches. Are they dealt with properly in context of swarm learning? Do we need some more work in those contexts? Does it depend on feature selection? Okay.
Okay, thanks. I'm trying to rephrase it. So you think of, let's say, nested batch effects that I have within an institution. So
Correct.
Yeah, let's say within day variation or experimentator.
Correct.
I think these kind of problems you also have when you are faced with just normal data integration. And what we have seen is that tools like SCVI are very much capable of dealing with this kind of nested batch effect. So they account for that internally. Everything else that I've seen that tries to account for, let's say, more than one batch covariate, let's say a day or experimentator, did not outperform this kind of, let's say, treat every batch equally for now.
I guess the difference is that in atlases, that information is typically available to us in terms of metadata, but in swarm learning, because of data protection and things like this, we may not know that there was actually a sub-batch per se. But thank you very much. That explains. Okay.
Right, so let's thank Maren and all of our speakers for a great session.